Let's continue our discussion about nutrient cycling. It's a really huge area in forest ecology. So many studies to talk about, and there's so much that we could talk about with every individual element, with interactions among elements, and this really makes up the field of biogeochemistry or forest biogeochemistry. But we're just going to touch on a few big concepts for Northwest forests today. First, let's think about mineral nutrition in plants. Mineral nutrients are elements like nitrogen, phosphorus, potassium that plants acquire in the form of inorganic ions. Nitrate and ammonium are the two major forms of inorganic nutrients that plants need, that plants use to acquire nitrogen. Nitrogen, of course, is critical for plant function, enzyme function, major enzymes like rubisco involved in carbon fixation. Phosphate is also really important, and phosphorus mainly is supplied to plants in the form of phosphate. And these tend to be growth limiting ions in forest ecosystems. Because there's a complexity to water movement, to carbon cycling, and nutrient acquisition, all of these things are tied together, and often the interaction of multiple elements, multiple nutrients, different forms of nutrients, ions, water flux, carbon flux, are all interpreted together in fields like forest biogeochemistry. When we think about nutrients, there are essential elements, and we can think about these as macronutrients and micronutrients. There are 16 essential mineral elements, and this varies a little bit by author, what is considered an essential mineral element, because you can find a lot of elements in plants that are more than just these 16. But these 16 are critical to plant function. We can divide these into nutrients that are required in large amounts and nutrients that are required in small amounts, though as we'll see, there's only a few that are really required in noticeably large amounts. So first, in terms of the macronutrients, we can take hydrogen, carbon, and oxygen and call those macronutrients, but really those are forming the backbone structures of organic molecules that form what we know of as life in vegetation. Then we have nitrogen. Nitrogen is abundant in, is probably the next most abundant nutrient, and is critical for enzyme function, like we said before, and more of what we consider nutrients when we're doing nutrient cycling research. Phosphorus is our next runner-up, next most abundant and most limiting to plant growth. And then we have a number of other macronutrients that are required, but they're not often in short supply. There are micronutrients additionally too, which are all required for healthy plant growth, though again, these may or may not be in short supply depending on the forest ecosystem we're working in. And then there are 4 or so other nutrients that are critical, potentially beneficial to plant growth, and some like aluminum, which can actually be toxic in high quantities, which is the case for some of these others too. Of course, you can find all of these on the periodic table, and so they're going to be familiar to you from there. If we think about concentration in dry matter, then it's really easy to see that these nutrients are not abundant in plants in the same concentration. So for example, carbon, oxygen, and hydrogen, those are all big components of plant matter. Nitrogen, okay, maybe a decent component of plant matter, 1 to 2%, somewhere in there. But then when we get below that, we have potassium, often around 1%, and then the other macronutrients in much lower percentages, often less than 0.5% plant dry matter. Phosphorus in particular can be limiting for plant growth, though it's less than 0.2%. When we think about what plants need, then you might think of plant deficiency guides or symptoms of plant deficiency. And so plants need more than Brondo, they need more than electrolytes. Plants need nitrogen, they need calcium, they need iron, potassium, they have all of these elements that they need. And when they don't have them, there are distinct symptoms of plant deficiency that we can read in plants. And so there are a number of these guides out there, and I encourage you to do searches to find them to help diagnose plant diseases when you find them, or plant deficiencies in nutrients when you find them. Additionally, soil pH has an effect on how available individual elements are in soils. And so under high pH conditions, alkaline soil conditions, many nutrients are less available than they will be under slightly acidic conditions. Most forests are in slightly acidic conditions, and so these elements tend to be more abundant. But it's important to know that these things can vary with pH. Plants or forests and forest ecosystems, and in fact, ecosystems in general, really differ in how they respond to limiting nutrients. So these data show bars for responses of different systems to addition of nitrogen, phosphorus, or nitrogen and phosphorus together. And as you can see, there are differences in different biomes with regard to how these ecosystems will respond to additions of these nutrients. So for example, in a grassland, we'll start on the far left, we might have a situation where there's a small positive response in growth to nitrogen alone or to phosphorus alone, but when you add them together, you get a synergistic effect, and there's a much more positive response when nitrogen and phosphorus are added together. So they're equally limiting, and together they produce much more biomass. Forests, you'll see, are actually pretty diverse. There are forests that show positive responses when nitrogen's added, positive responses when phosphorus is added, much more positive, and sometimes a similar response when they're added together. And so there's a variety of responses across different ecosystems. Nitrogen tends to be limiting to many ecosystems, whether it's a large limiter or a medium limiter, and especially in temperate forest ecosystems. Nitrogen tends to be more limiting than phosphorus or other elements. There are some studies suggesting calcium can become limiting, particularly with repeat harvests of forests, but nitrogen is almost uniformly limiting. This is ironic because nitrogen actually forms a large percentage of our atmosphere. We have a lot of nitrogen. It's unusual on our planet to have such high nitrogen abundance in our atmosphere. We're unusual compared to Venus and Mars, where we have about 80% of our atmosphere is nitrogen. Yet it's impossible for us to, or it's very difficult for us to get and for plants to get. And this is because nitrogen is triple bonded. We've talked about this in earlier lectures. It's very hard to break that apart. So to help, let's look at a diagram of the nitrogen cycle. Okay, that might be too simple of a diagram of the nitrogen cycle. Let's look at another diagram of the nitrogen cycle. If the first one was too cold, then this one might be too hot. There might be too much going on here to understand what's happening in the nitrogen cycle. Let's see if we can find something just right. Okay, we're getting a little bit better. Here's another diagram of the nitrogen cycle. We have atmospheric nitrogen. We have nitrogen fixing bacteria that take it up. Somehow this diagram only has nitrogen fixing bacteria in legumes. We're going to learn they're not just in legumes, they're also in the soil. So that's good, that's there. There are also some others that we're not talking about here yet. But we have nitrogen fixing bacteria taking N2 gas, turning it into ammonium, then turning that into nitrates, and nitrates are suddenly taken up by plants. Okay, there might be a few more steps here. In particular, we have other species that can pull in nitrogen. We have plants that can take up ammonium, but we're getting better here. Let's see what our next. These are the two main genera involved in nitrogen fixation and symbiotic relationships with plant roots. And so let's try this one. Okay, this one we're getting better. So now we have, let's see, a reservoir of nitrogen in the atmosphere. Okay, looks good. We have nitrogen fixation. Oh, now it's looking better because you know what? Turns out nitrogen can be fixed not just by microbes, but also by lightning. So we have lightning represented here. We also have some wet deposition of nitrogen. So that's good. Nitrates end up in the soil. Okay. Fertilizers are added here. Oh, that's right. We can add nitrogen through fertilizers and artificial fixation of nitrogen from the atmosphere. Okay, then we have fixation of nitrogen by nitrogen-fixing bacteria. Of course, we've got them in legume root nodules. We also have them in alders, which we'll talk about in a second. Okay, I like this one a little bit better. A key thing I like about this diagram is that at least I've got a transfer of nitrogen among sources from ammonium to nitrates to nitrites being taken up by plants. But I think we can get better still. Okay, here we go. Okay, so there are so many diagrams of the nitrogen cycle available online. You're going to find all sorts of diagrams, but they're not all technically accurate. Sometimes they're just trying to emphasize one component of the nitrogen cycle over the other. The nitrogen cycle is a complex cycle, and it's important to get all of the components right if you're trying to understand forest ecosystems. This artistic rendering actually does a pretty good job. So we have a high amount of nitrogen in the soil. Back to that. Nitrogen can get into the soil a few different ways. It can actually get in through lightning, believe it or not. Lightning can result in nitrogen fixation that then can be in a breakup of that triple bonded molecule N2. We can have bacteria that are able to fix nitrogen, either in the soil or in association with plants in root nodules. This can take place in legumes for sure, but there are also other species like ceanothus and alder that also are involved in nitrogen fixation. We also have anthropogenic means by which nitrogen can get fixed, like the Haber-Bosch process. We'll talk about that in a second. High pressure, high heat can break apart that triple bonded molecule and fix it into um, uh, inorganic forms. Okay. So once nitrogen is added, it can be added through fertilizer, through bacteria in association with plants or free living in the soil, or through rain and dry deposition. Once that happens, then the biological components of the nitrogen cycle become really, really important. So nitrogen can get fixed as ammonium. That ammonium can result in nitrogen that's available to plants in a form that's available to plants, or microbes can go through and nitrify that, and we can have nitrification that results in nitrate. Nitrate can also be a form of nitrogen that's freely available to plants, but it's also available to microbes. And so at this stage of the nitrogen cycle, plant interactions with microbes are really important. Um, Depending on conditions in the soil, we can have denitrification processes where nitrogen is turned back into nitrogen gas in the atmosphere. Additionally, buildups of high amounts of nitrate and ammonium in the soil can lead to eutrophication, where that nitrogen is leached into groundwater and can actually be a major pollutant for water systems. So a lot of complexity here. The Haber-Bosch process is a really important nitrogen fixation, a route by which nitrogen gets fixed into inorganic form. This is a process for ammonia synthesis. And so you take high temperatures, you take high pressures. So we're talking up to 550 degrees Celsius. We're talking pressures up to 350 atmospheres. And in this industrial process, you can take N2 gas and fix it into ammonia. This takes up about 1% of the world's energy, and this is really responsible for the Green Revolution, or what people have called the detonation of the population bomb. In other words, a large amount of our current human population can be accredited to the invention of industrial fertilizers, and in particular, this process, what's known as the Haber-Bosch process. Here's world fertilizer use going from 1960 to 2008, and you can see how that has just grown tremendously. Along with that comes patterns of ammonium deposition. So when you have artificial fertilizer created, you also create a route by which that fertilizer can get blown around by winds. It can get transferred into the air and then deposited in other regions. So in the agricultural heartland of North America, in the US, we tend to have very high amounts of ammonium deposition. when you measure ammonium deposition in the air, and that's associated with agriculture and fertilizer use. That has a big effect on forest ecosystems for sure. Here's an example of wet, dry, and cloud water deposition of nitrogen in a variety of ecosystems. So these ecosystems are taking place in different locations, and the abbreviations at the bottom stand for the name of those different ecosystems. Klingman's Dome in North Carolina is CD, Pawnee, um, uh, Colorado is PW, Whiteface Mountain, New York is WF, Coweta in North Carolina is CW, and on and on. So we have a number of different locations here. OR doesn't stand for Oregon, by the way. This is Oak Ridge in Tennessee. The point of this figure, though, is just to show you that the ratios of wet and dry deposition, that is, in this case, nitrate being deposited in dry air through dust, or wet deposition through rainfall, or cloud deposition, really varies by system. And some systems really have high amounts of deposition of nitrogen. That's going to play with the nitrogen requirements of those systems. So when we think of forest ecosystems anymore, especially with high nitrogen deposition rates from the atmosphere, we can no longer think of them as closed systems. Anthropogenic nitrogen fixation has a big role in influencing how systems are limited in terms of their nitrogen. And if it's being transported around via rain or wind or dust, then we can have some big alteration into historic nitrogen limitation scenarios. However, all that's aside, we can still think of Northwest forest systems as mostly being limited by nitrogen, but it's good to be aware of this stuff. Let's talk about some natural ways that nitrogen gets into systems. In many of the nitrogen cycles we just looked at, there was a step where nitrogen fixing bacteria had an association with a legume root or a nodule. Well, it turns out this can also take place in one of our common riparian forest tree species, red alder. And so red alder is one of these 4 tree species that are commonly found along riparian areas or streams in the Northwest, a tree species that's often dominant and a really important one for thinking about nitrogen. It often forms monotypic stands, not because it's clonal, but because it has really small seeds that disperse and take advantage of low nutrient conditions in highly disturbed environments. It's in the family Betulaceae. It's probably the most common hardwood in the Pacific Northwest. Okay, maybe bigleaf maple might be a competitor for that title, but regardless, it's pretty common in the Northwest. It's short-lived. It doesn't tend to last very long. It's not a very shade-tolerant species. It has rapid juvenile growth and then slows down fast. It tends to rot fast. It was regarded as a junk tree for a long time in the forestry profession, but now it's considered valuable. People use it for trim and all sorts of interior work. Well, before we talked about nitrogen fixation requiring high amounts of temperature and pressure in order to get it to take place in an industrial context. But it turns out that the nitrogen fixing Actinomycete frankia, which forms nodules in the roots of alder, can do this for free. It lives in symbiosis with alder and benefits from sugars that alder will give it from photosynthesis. And in return, in these little tiny nodules, it will fix nitrogen out of the air and deliver it to alder. And so then alder gets a free supply of nitrogen, which can really transform the requirements for growth for that species, especially when nitrogen's limiting. Well, there are a couple other really important and interesting routes by which nitrogen gets into systems in the Pacific Northwest that differ from other systems. And one of them is through nitrogen fixation by arboreal or canopy lichens. So Lobaria oregana in particular is a species that is known for its nitrogen fixation. I think the image we have here may be a little bit green for Lobaria oregana. There we go. That's a little bit better. Okay, so now we're looking at Lobaria oregana. Um, and let's talk about what this species does. So here's an abstract from a paper. This is by Marie Antoine, one of a number of researchers who've really focused on this species. And this is from a paper that's actually quantifying the nitrogen fixation. And so this is at the Crane site in Wind River, where they estimated annual nitrogen fixation at 1.5 kilograms per hectare. And then in the experimental H.J. Andrews experimental forest, it might fix at least twice that, if not 10 times that. So depending on the system, the amount of nitrogen fixed by this canopy lichen can be pretty variable. Here are a couple of images and a table from a number of different sources. And in this case, the first upper left panel is talking about a second growth forest that's undergoing stem exclusion. And in that stem exclusion phase, we might expect that there's a lot of competition for something like nitrogen. When we compare nitrogen accumulation in different systems, intact forest covered by Douglas fir, like the stem exclusion phase forest, or red alder stands, we would expect really big differences in nitrogen cycling. And in fact, we find that. So for example, in this table, you can see that nitrogen accumulation, if the overstory is Douglas fir, is about half of what happens in the overstory of red alder. Understory can be 10 times higher under red alder. Forest floors can be many times higher under red alder. Soil can be almost twice, can have almost twice the amount of nitrogen in soil under red alder as compared to Doug fir. And so alder is clearly plays a big role not only in gathering that nitrogen up, but contributing it to the ecosystem. The same is probably true of Lobaria and other species that fix nitrogen in ecosystems. Here's a study from H.J. Andrews again. Now, this is from Watershed 10 in H.J. Andrews. And this is a really interesting table because it's showing us not only nitrogen, but potassium, calcium, phosphorus, and organic matter. It's showing us the amounts of these materials in different components of the forest overstory. And then it's showing us per year incremental increases in these elements per year, or how much is needed per year incrementally. And then it's showing us fluxes, including fluxes through atmospheric precipitation, atmospheric particles, overstory litter fall, leaf wash, stem flow, and leaching. Interestingly, the stand where these data were collected, I believe this is one of the forests that recently burned in the 2020 fires at H.J. Andrews. What you should pick out from here is that the amounts of nitrogen and these other elements are not all equal. And so the amounts of nitrogen, for example, present in the soil rooting zone are much greater than the amounts of these other elements. And the incremental requirements for nitrogen are much higher than for other elements. So not all elements are equal. Microbes can play a really big role in regulating nitrogen availability, and that's easy to overlook. Let's talk about a method by which we measure nitrogen availability, and then we'll come back to this. It's really important when considering nitrogen to realize that nitrogen availability is dependent on fluxes of nitrogen between pools. It's really dependent on rates of mineralization, but it's not as dependent as you would think on the total amount of nitrogen in a system. So when we're talking about nitrogen availability, we're not talking about how much nitrogen is in the system. We're talking about how much is available for plants to take up. And that's very different than the total amount that's present in a system. So if we were going to measure nitrogen mineralization, then one way to do this is to use a core technique where we have cores with walls that don't allow plants to take it up. And if we installed 3 cores in a forest soil on the top panel, we might immediately pull one core and measure how much nitrogen was present in the soil at the time we installed the cores, and leave 2 cores to incubate in the field, one with a lid on it and one without. We could come back, say, 6 months later. This would be during the growing season. We did this at Evergreen in 2011, which is where this figure comes from. 6 months later, we came back and extracted the tubes that are the black tubes there that were incubating in the soil. Now, one was covered and one wasn't. And the reason for that is that the covered one blocked any atmospheric inputs and the uncovered one didn't. The difference between those two will allow us to determine how much nitrogen is brought into the soil through atmospheric deposition. The reason that we're incubating the soils out there is that we're incubating them in a chamber that walls off the ability of plants to take up nitrogen from the soil. So nitrogen that isn't taken up by plants will just accumulate in the chamber. This is important because microbes are continually turning over organic material, turning organic nitrogen into inorganic forms that normally plants would take up. If plants aren't there to take it up because they're walled off by a chamber, that nitrogen accumulates. And over time, resampling, we can see how much nitrogen accumulates and how much would have been taken up by plants. In other words, we can see how much available nitrogen there was, how much nitrogen was made available to plants that they could have picked up. So if we take our incubated core and we subtract off our initial, then we get net nitrogen mineralization plus deposition. But we can get the total amount of nitrogen that was available for plants, say in a form like nitrate or ammonium, which we can measure directly, and we can measure using instrumentation. If we take our covered core and compare it to our initial, we can get net nitrogen mineralization, the amount of nitrogen that was liberated from organic form into inorganic form that would have been taken up by plants, but wasn't. So that's our net nitrogen mineralization or nitrogen mineralization rate. This is generally calculated as the combination of nitrate and ammonium in forest soils. And then if we take our incubated soils and we subtract off our covered incubated soils, we can get net nitrogen deposition. Here's yet another way to look at the nitrogen cycle. Like I said, there are a million of these graphs out there, or these figures out there. In this one, we start with soil organic matter, and soil organic matter gets decomposed and we end up with dissolved organic nitrogen. That dissolved organic nitrogen can actually be taken up by the plant. So yet another complexity that wasn't in our previous graphs. More commonly though, that dissolved organic nitrogen gets turned into ammonium. And so there's ammonification of that material, resulting in the inorganic ammonium form of nitrogen. That can be taken up by the plant. It can be taken up by mycorrhizal fungi. It can be brought to the plant. It can also get taken up by microbes, and microbes tend to have first choice. It can also get turned into nitrate. So we have nitrification that takes place. And once it gets turned into NO3 negative or nitrate, then it can again get taken up by the plant. Nitrate tends to be more mobile than ammonium in forest soils, and so it's more likely to be leached out. At especially the stages where it's turned into ammonium and NO3 negative nitrate, we have the potential for gaseous loss of nitrogen back out into the atmosphere. And so that's important to know. Here's another way of thinking about it. We have processes, reactions, and optimal environments for all of these things to happen. So if we start with a dissolved organic nitrogen and we go towards ammonium, then we go towards nitrite and then nitrate. NO2 negative is nitrite, NO3 negative is nitrate. That's a nitrification process. The process of taking organic nitrogen and turning it into ammonium is the ammonification process. And then following that, we can have a denitrification process if we're in a low oxygen and high carbon environment. Forests get discussed a lot with regard to site treatment and fertilizer application. This is a graph from a textbook by Chapin et al. In 2011. And this graph does a really nice job of showing nitrogen and phosphorus concentration in inorganic and organic forms in different ecosystems with different amounts of forest cover or range cover or agriculture. And what you'll notice here is the order of magnitude difference in the amounts of nitrogen and phosphorus that are present in agricultural systems that are over 90% dominated by agriculture. So forests get talked about a lot with regard to fertilizer inputs, but the amount of nitrogen and phosphorus present in both organic and inorganic forms in agricultural systems can far outweigh what we see in forest ecosystems, regardless of fertilization activity. Temperate forests tend to be limited by nitrogen, and we've talked about that before. And so we know that adding nitrogen can certainly boost production. And that's certainly true in the Northwest. We have a lot of private lands in the Northwest which apply nitrogen. And the application of that nitrogen has big consequences, both for forest growth and for greenhouse gas emissions from those ecosystems. So playing with the nitrogen cycle by adding nitrogen can have a lot of unintended consequences. And one of them is the release of more greenhouse gases from ecosystems that are fertilized. But it's undeniable that it increases productivity. There are also, just like there are trends through time in carbon accumulation, we can also think about trends through time in nitrogen accumulation in topsoil for typical Northwestern forests. So in this figure from Pearson Education, we have pioneer alder stages increasing nitrogen until we get to kind of a transition, and then in this case, a later, spruce stage for this ecosystem. But you can think about similar trends in our Douglas fir forests of Western Washington, where intermediate alder stages might have higher nitrogen in the soil that declines as that forest goes towards old growth. Similarly, after fire, we know that there can be immediate release of nitrogen in some forms immediately after fire, but in general, a lot of the nitrogen that is liberated in a fire can get leached away or can be volatilized and disappear. And so because of that, systems generally are expected to increase in nitrogen through time following something like a fire. And organic matter, as we know, accumulates through time. Organic matter has high cation exchange capacity, but it also contains a fair amount of nitrogen. So often nitrogen will track trends in carbon through time, where other cations and other soil properties may not. This illustration is showing some critical elements and their cycling in a simple form in 4 separate panels. So first, in the upper left, we have the nitrogen cycle. Now, we've seen all sorts of complex nitrogen cycles in this one lecture from all over the place. And so here's a nice simple one for you to chew on. Nitrogen comes in, gets converted to ammonium, that can be taken up by plants, gets converted to nitrate, that can get taken up by plants or leached. And then both of those can potentially degas back into nitrogen gas in the atmosphere. There's also the potential for nitrogen to go into dissolved organic forms with different fates. Importantly, when nitrogen is taken back up by the plant, a significant amount of the nitrogen cycle, and this is a big take-home point, a significant amount of the nitrogen cycle is internal cycling. And so nitrogen is taken up by the plant, it's dropped in the leaves, it's decomposed, made available to the plant in inorganic forms, taken up by the tree, dropped by the leaves, decomposed, and taken up, and on and on and on. That cycle goes on. The inputs and the outputs in terms of nitrogen fixation and the outputs in terms of degassing of nitrogen are relatively rare events as far as the nitrogen cycle is concerned. Those are small amounts of small fluxes. The big fluxes in the nitrogen cycle, typically in a forest ecosystem, take place internally. The same can be said of phosphorus, but in the phosphorus cycle on the upper right, the big difference is that this is a much simpler cycle in the sense that phosphorus is originating in rock. We've talked about phosphorus being broken down, not just physically weathered, but chemically weathered into phosphate, which is available to plants. Then there's a tight cycling of phosphorus within the plant soil ecosystem. So the plant takes up phosphate, it releases phosphorus in organic form, that's mineralized back to phosphate, which is taken up and then released, taken up and released, and the cycle continues on and on and on. And there's relatively small amounts of leaching from the system, represented as E here, and small amounts of inputs of phosphate into the system through time. So most of it, again, is internally cycled. A couple other diagrams here for sulfur and potassium, which are also interesting figures to chew on. A couple other points that I'll leave you with before we're done with this lecture. We've seen this figure many times before. This is a real model for phosphorus availability and thinking about how phosphorus cycles in ecosystems worldwide, but especially in forest ecosystems. And so this is similar to what we were just talking about. It's important to know that physical weathering of phosphorus isn't enough. It needs to be chemically weathered into a form that's available for plants. That tends to increase through time, but the availability, total availability of phosphorus in an ecosystem declines through time as phosphorus is weathered out of the system. Soil conditions can also really affect the availability of something like phosphate. So phosphate tends to be most abundant around a pH of between 6 and 7. And then depending on reactions with other components of the system, phosphate can become less available. It can be fixed by hydrous oxides of iron, and aluminum. It can go through fixation as calcium phosphate, and there are all sorts of other reactions that can also take place. So available phosphate is really different. So just like nitrogen, we're not totally as concerned about the total amount of phosphorus in the system when we're thinking about forest nutrition. We're thinking about the amount that's freely available for plants to use, and that depends on other soil conditions and other biogeochemical reactions in the system. So total phosphorus is not the same as available phosphorus, just like total nitrogen is not the same as available nitrogen for plant growth. However, you can get a really interesting signal when you look at plant growth itself. So the ratios of phosphorus content to nitrogen content in plant material can tell you about the relative limitation of nitrogen and phosphorus in forest vegetation. So for example, in this image, the black dots represent nitrogen fixation for sites that have where you have vegetation that has higher amounts of phosphorus relative to nitrogen. On the flip, the white dots represent phosphorus limitation scenarios where you have high amounts of nitrogen relative to the availability of phosphorus. Remember that there are biological reactions that require these things. There are biological building blocks that require these things in certain ratios. So it's not enough just to add nitrogen endlessly and have a forest continually grow. It's always going to be limited by something. And so a forest that's no longer limited by nitrogen because it has all the nitrogen at its disposal it could possibly want will then be limited by phosphorus because it's not just one thing that limits a system, it's multiple things can limit a system at the same time. These ratios tend to be pretty consistent though, and that's what's really fascinating, is that there's some consistency in the stoichiometry of these elements in components of plants. So in this case, plants with an N to P mass ratio of less than 14 or a molar ratio of 31 respond primarily to nitrogen, where plants with nitrogen to phosphorus mass ratio of greater than 16, which comes out to a molar ratio of about 35, respond primarily to phosphorus. And this again comes from that Chapin textbook. Okay, one last figure I'll leave you with. One of the classic types of ecosystem studies that have been done in forest ecology, research, is to do whole watershed experiments where things are done to an entire watershed, and then you can look at what comes out in the stream water as a result of that watershed activity. We talked about some of that with regard to H.J. Andrews and some of their watershed experiments with logging, presence of roads, not presence of roads, old growth forest versus young forest. In this case, we're looking at results from the Hubbard Brook ecosystem in northeastern US. This is a classic system where some of the first biogeochemistry studies that were done on this scale were conducted. Um, this graph is showing losses of calcium, potassium nitrate, and particulate organic matter in stream water before and after deforestation of an experimental watershed there. The shaded area shows the time interval during which vegetation was absent due to cutting of the trees and then application of herbicides to keep plants from growing. And then you can see what happened after a reforestation was allowed. So we start with a 60-year-old forest. There's a cutting event. Reforestation is prevented. And notice how nutrient loss from the system just spikes for calcium, potassium, nitrate, and actually eventually for particulate matter too, when vegetation isn't present. This was taken at the time and is still taken to indicate the amount of uptake of these elements by vegetation in the system. And so when that vegetation isn't present or when the system is cut, then there's a significant export of this material out of the system when it would have previously been held by the vegetation or taken up by the vegetation. And so in recovery of vegetation, you see that export or the nutrient loss goes down rapidly. And there you have it. It's what a forest does for you. Okay, that's it.